Hey, welcome everyone. Thank you for coming. We'll start on top of the hour. I shared the folder. There are all the links for all the resources we will use today. There's a PowerPoint presentation and a few movies that the guest speaker will use for the presentation. Feel free to check them out. I'll share in the chat also the publication. So we'll start in a little bit. Thank you.
Hello.
Hi, how are you? I uploaded
Good.
also the paper into the folder and everything, so it should work. You can try it out if you would like.
Okay. Uh, let's see. Yeah, I see the paper. Cool. So I removed all the movies from the um, presentation and I just put files instead. Okay.
Yeah, I saw. Yeah, thank you for doing that. Yeah, thank you. We'll
I'm gonna
start
see if I can
in
see the.
like around 8 minutes, so we still have a little bit of time. I'm muting because I'm inviting people in, like I'm pinning them in
Okay.
and so on. So and then I'll share on Twitter really quick also that we are about to start. So
Mhm.
okay. Hey everyone, we will start in around 2 to 3 minutes. So, if you think this discussion will be interesting, feel free to share the room with people. And yeah, all the resources that we will need today for the talk are in this Google Drive folder that I posted above. And in the chat, I shared the lab website. And yeah, thanks for coming. And we will start soon. I know people will still be coming in when we start, but yeah, and welcome, Thierry. Thank you so much for making the account, coming here, and
Thanks for
we
having me.
appreciate it.
Can you hear me well?
Yes, I can hear you well. Thank you.
Okay.
And we still have like 1 or 2 minutes, so yeah. Anyways, but we'll start soon. And I hope you had a good day. I know we had a lot of trouble with the movies, but
Yeah, I think I removed the movies from the slides, and so people should be able to just click on the movie if needed.
yeah, and so usually, like, because we keep the recordings, we keep also the files up. Like, I have enough data space to keep them up. So if people listen in a maybe in a few days, the recording, it will still. If you want at some point me to take it down, just let me know and I'll take it down.
Mhm.
Yeah. Okay. Um, yeah, we are about to start, everyone. So thanks for coming. And yeah, as I said, people will still be coming in, but I shared also in the chat like where to find everything and so on. So when we start, people know by then. Okay, I think, hi Dr. Shah, welcome. Thanks for coming. Meet Thierry. Thierry, meet Dr. Shah.
Hello.
Hi, Katerina, and our guest here. Hopefully, everyone had a great day so far.
I'm glad to hear. Okay, we are starting right on time. Perfect. So, welcome everyone to Science Society, and of course, a special welcome to you, Terry. We really appreciate you coming here. And before we start, let me introduce you a little bit to the audience so they get to know you a little bit. So Dr. Thierry Emonet is a professor of molecular and cellular and developmental biology and physics at Yale University, and he does really interesting research in combining mathematical modeling and quantitative experiments to understand biological computations that allows organisms to sense and navigate their chemical environments. And he uses model systems in his lab, um, bacterial chemotaxis and fly olfaction, and they can then compare the different models, which is a really interesting approach. Before coming to Yale, Thierry studied physics at the ETH Zurich, where he received his PhD cum laude in theoretical astrophysics from the University of Laguna. And before doing his postdocs at the National Center for Atmospheric Research, Boulder, and the University of Chicago, where he discovered key mechanisms that enabled magnetic field to float to the surface of the sun to create sunspots, which is really interesting too. Maybe we should talk about that another day too. Supported by major funding of NIH, NSF, and the Paul G. Allen Family Foundation, and many more. And he is also very interested in art. And actually, his wife is a renowned sculptor, which is really interesting. I checked out the work; it's really interesting. So, welcome. And we usually start.
Hello?
Um, so, how did you discover your passion for science? Was it something you always wanted to do? Did it come later? Did you first want to become an artist? Maybe? I don't know. We really always love to hear like the story of people, how they became to be, the person in the career they are right now. Thank you.
Oh, thank you so much for having me. Um, I was having trouble at some point. The connection was dropping, so I don't know if it was me or you, but I really I heard everything more or less what you said, so it's okay. Um, the so where did I get how did I get in science? So I grew up in the mountains of Switzerland as a kid, and basically my passion was just rock climbing, and I also liked mathematics, and I didn't really think so much about science. I was just really enjoying, uh, rock climbing those mountains. But I discovered like a really a great taste for like studying physics and understanding nature and so on. And so I went to the ETH and then to the Canary Island to study astrophysics. And when I then moved to the US to do a postdoc, first at Boulder and then at the University of Chicago, um, at that point, I met this person who had just been hired as an assistant professor at the University of Chicago. His name is Philippe Cluzel. And he told me, in my lab, what we study is where does individuality comes from? Why are we different? And I studied the individuality of bacteria. And so he was basically tracking the behavior of individual bacteria as they're navigating chemicals and studying the fluctuation in the behavior of those bacteria. So I thought that that was really fascinating, and this is how I went on to to switch to biology. And with respect to art, I always had like this thing inside of me. So at some point, I really considered seriously to become a sculptor, but I ended up marrying one, which is pretty nice too. Can you hear me, by the way? Since I don't get feedback, so I
Oh,
don't know
yes.
if
Yes,
it's
yeah, I can hear
yeah.
you well. I'm sorry, I
Okay.
had the red bar in between. I think it's because I went to another room further away from
Oh,
the.
I see.
Um,
Okay.
yeah.
So the first thing I wanted to tell everyone is that please interrupt me right away with any questions so we can have more of a conversation than me just talking to a screen here because I don't see you, so I don't know if you're still there or not, and so on. And maybe I can just start telling you about chemical sensing and how did we get to study how flies navigate odors.
Yeah,
Uh,
sure. Thank you.
so I'm bringing up my slides on the phone, and I was not able to embed movies. So whenever I mention a movie in the slide, you're gonna see movie one or movie two. That means you can go to the Google Drive and click on that particular file and to visualize it. So the work I'm going to talk to you about was spearheaded by an amazing postdoc, Nirag Kadakia, and he was also collaborating with another postdoc, Mahmoud Demir, and this is a collaboration with my colleague Damon Clark, who happens to be an expert in motion detection in the visual system, and you're going to see how that comes into play. So navigating chemical is really something that's essential for life. You cells follow chemicals, bacteria follow chemicals, insects follow chemicals, animals, etc. And as far as animals, olfaction is really the primary sense that they use for survival, to find food, find place to lay eggs, to find sexual partner, etc. But also to navigate, to find you, to pinch you if there are mosquitoes, spread disease, etc. No? But it's also one of the sense that's, in some sense, less understood for two reasons. First, it's very difficult to visualize odor in space and time. They're not, visible molecule. And the second thing is odors are made out of many compounds, odorant, individual molecules, and it's the combination of those that give you the sense of a certain type of odor. And how is the odor identity is represented in the brain is something that is still kind of mysterious. But so today, what I'm gonna talk about is not so much about how insects and animals recognize which odor they are encountering, but how do they follow those odors to their sources. For example, if it's a pheromone released by a female, or if it's some odor that's released by food that they'd like to get to. And so if you go to the third slide, basically the main question I'm interested in is what strategies do organisms use to navigate chemicals? Strategies in terms of what do they need to measure and how do they need to use that information in order to get to the source of odors. And so as a first example, if you move to the fourth slide, there's a slide about what bacteria do. So bacteria, these bacteria, there's a movie. No, I don't have the movie in the drive. But basically on the left, what you're seeing is E. coli bacteria that are swimming. And that's a movie that was taken by Howard Berg from Harvard. And you see that there's the body of the cell in the front, and then there's this kind of wavy thing in the back. These are the flagella of the bacteria, and the bacteria spin those flagella, and when they spin them in the counterclockwise direction, the flagella form a helical bundle, as you can see in that picture, that pushes the cell straight. And whenever they spin the motor in the other direction, that helical bundle becomes unstable, and so that causes the flagella to kind of fly apart and cause a sudden change in orientation of the bacterium, and this is called a tumble. So on the right of the slide, you see this diagram showing that the bacterium is actually doing a random walk, and it's doing a random walk because it's so small. It's about 1 micron to 2 micron long, and over this distance, small molecules diffuses extremely fast in 10, 15, 20 milliseconds. Molecules will diffuse from the front to the back. And so if the bacteria don't move, they're not able to tell in which direction the concentration is increasing. So what they do is that they need to move in a given direction to see if life is getting better, which means if the attractant concentration is going up or down. And if it's going up, then they reduce the probability to change direction. So they have a sensory system that controls this probability to change direction, and they reduces that. But all of this work well if they are in a gradient that is stable. And so in this picture, the gradient that's stable is represented by that smooth blue color that's going from, white on the left to darker blue on the right. No? And if you go to the next slide, bacteria are not the only one who climb gradients. The larvae of the fruit flies do this. This is a figure from a paper from Mathieu Louis' lab, where they examine how fruit flies navigate towards the source of odor. And the same thing, you can see that the odor concentration is this smooth landscape, And so the larvae can tell if it's going up or down that landscape, and it can also sweep its head when it's stopped to tell, oh, the concentration is higher on the left or on the right, so I should go on the left. Is that clear? Is there any question up to here?
For me, it's clear, and I don't see any comments or questions in the chat.
Okay.
Thank you.
So then the question is, now imagine the same situation, but you add wind. So now there's a source of odor, but you have wind. And so the wind is going to carry those odor molecules downwind. And if the wind is a bit turbulent, the signal is going to be very complicated. And so if you want to go to the next slide, you'll see this picture with trajectories that have different colors, and there's a dark kind of image of a plume in the back, and it says movie 1. So if you click on the first movie, you can visualize it, and we can talk about it a little bit. And so I'm gonna turn it on my computer. So tell me if you are able to actually visualize that movie.
I'm good. Is everyone else okay? Or if not, please share in the chat or raise your hand. Thank you.
So if you're able to visualize it, you'll see that basically you have a few flies that we're tracking. And what's happening here is that those flies are forced to walk between two pieces of glass. So the glass are about 1 centimeter apart from each other. And so they can walk on the surface of the glass at the bottom. But from the left side, we're flowing in air, and in the middle, we're releasing a plume of smoke. And it turns out that the fruit flies love smoke. And so they track the smoke towards its source. And so you can see their behavior. And this was a very nice discovery that one of my postdocs, Mahmoud Demir, made, that basically using this assay, he could see the full behavior of the fly as a function of time and the statistics of the encounter of the fly with odor packets that are discrete. And you see that unlike the case of the bacteria I was talking to you about, the larvae, there's no smooth concentration of the odor. What you see here is like discrete packets that are dragged down by the wind. And what's happening is basically the wind is in this case is not turbulent, but we basically perturb the wind with lateral jet to make it to approximate turbulence. And this basically rips this straight plume that you see at the beginning into different pieces. And so an animal in the middle of that area only experiences intermittent whiffs that are odorized, and then a duration of time when there's no odor, and then again some odor, and then no odor, and so on. No? And so the question is, how do you navigate when there's no continuous signal like that, but you're just basically experiencing these very intermittent packets of smell? Any question?
I think we're good on the questions right now. Thank you. But yeah, it's an interesting question you ask.
So I want to make you guys work, but I don't get very. So far, I haven't managed to do it. But so the next question then is, what should be the strategy that insects use to navigate such complicated signal? And this is a question that has been around for more than 100 years. So a lot of scientists have been pondering about this. And there's a beautiful experiment So if you go to slide number 9, the one that on the title says Kennedy and coworkers. So what Kennedy and coworkers did is that they realized that one of the main problem was it's very hard to know where the odor is. And so if you want to understand how the animal navigate the odor plume, somehow you have to know where the odor is relative to the animal. And so they did this cool experiment. They released pheromones together with soap bubbles. And then they released a moth downwind from that location that they knew was attracted to this pheromone. And they basically recorded the trajectory of that pheromone. And because they had the soap bubble, they knew when the animal was inside the odor plume or outside of it. Okay? And so the diagram that you see here is a trajectory of a moth. And the line is thicker when the moth is inside the odor plume and thinner when the moth is outside the odor plume. And what you realize, what you see here is that whenever it's thick, the moth seems to be going upwind. And whenever it's thin, when it loses the odor plume, basically the moth starts to go crosswind. And so that suggests that a very basic strategy to navigate this intermittent signal is to go upwind when there's odor, but go crosswind or casting crosswind to try to find the plume again until they intersect with the plume again, and so then they go upwind again. And so this, in this strategy, if you go now to the next slide, wind provides the directional information, and the encounters with the odorized air packet provide timing information. Tells the animal, oh, you lost the plume, you better go and start to cast, or basically tells the animal when to cast or when to go upwind. But the directionality, the direction information is provided by the wind. Okay, and since the 30s until the year 2022, this has been the case. Basically, that the main directional information that is available to animal to navigate is the wind. And so the question we ask, which is the next slide, is the wind really the only local directional cue available to navigate turbulent plumes? Is there a possibility that those animals might also detect something, another directional cue that might help navigate? And so one of the inspiration to ask this question came from basically my path in astrophysics. So if you go to the next slide, you see this beautiful picture, which is actually on the cover of this amazing book. I really recommend to all of you, if you can get your hands on a copy, you should get it. It's called An Album of Fluid Motion. And it's basically a compilation that Milton Van Dyke did of fluid mechanics experiments. So they're basically pictures of fluid mechanics experiment. So in this case, for example, you see there's a cylinder on the left, and there is a laminar flow flowing from left to right. And on the cylinder, there is some dye that's being released. And you see behind the cylinder that there's this wake of vortex or vorticity or swirling motion of the fluid that is dragging that tracer that's being released from the cylinder, no? And so this is not a turbulent regime. So there's a regularity to it. But what you can see also is that when you go from the cylinder downstream, these volutes become larger and larger. You see that? So on
Yep.
the right, they're much more broad. And so that means that there's some, in addition to going downstream, these tracers are moving outwards from the center line. And so if you go to the next slide, it's also a picture that's in that book. This is in a regime that it's much higher velocity, and so it's in a turbulent regime. So now you see again these packets of this tracer are broken up, no? But again, you see that there's this spreading of the signal away from the center line. And this is one of the universal effects that turbulence will have on passive tracer. So turbulence will cause those, by breaking up and twisting and so on, those packets of tracers, they basically cause those packets to perform a random walk, and this random walk disperse the signal laterally away from the center line much, much faster than this would happen if you had just molecular diffusion. So if this flow was just laminar, you would just see that the plume would be straight and slightly wider on the right than on the left due to molecular diffusion. But the turbulence caused this dispersion that is very fast in the lateral direction. Okay? And so this is present almost always when you have an odor that's being released in air. The air has this complex turbulent motion, and that will basically disperse the signal in addition to dragging it in the main direction of the wind downstream. And so one thing we wonder was, well, there's this regular motion of those packets, not only downstream, but also sideways, no, away from the center line. So if you could detect that, you would have an extra information. You would, if you could detect the red arrows as an animal, you would know not only where is upstream, but also where the center of this plume is, which would be a very valuable information to have. No? And so if we go to the next slide, I'm showing like a simple simulation of a theoretical model of this dispersion. So here we basically approximating this by taking a model where you have a source that's releasing packets at a random rate, at a rate constant, but randomly. And those packets are dragged downwind, but they're also performing this random walk due to this turbulent motion of the air, no? And in addition, they're also diffusing. And so those circles, you see that they're becoming a bit wider as they're going down to the right. That's molecular diffusion, but also they're spreading much faster than they're getting wider due to this turbulent diffusion. And so if you calculate the mean wind in this simulation, the mean wind direction is basically straight from left to right. But if you calculate the odor velocity, so basically you find that there's a field that is pointing outwards. Okay, and by the way, this understanding this dispersion was beautifully described about exactly 100 years ago by G.I. Taylor. He's a fantastic physicist working in fluid mechanics. Okay, so these consideration brings up on the next slide, slide 15, the hypothesis that maybe flies could detect the direction of motion of odor packets independently from the ability to detect wind. And this is important, and I will see why. And so detecting the direction of motion would be very useful because there's more regularity in that direction of motion than you would have, for example, if you were to just try to use the direction of the gradient. For example, in this picture here that you're seeing now, the blue arrows are basically the direction in which the gradient of the odor is pointing. And you see that it points in kind of random direction. But the direction of motion of the odor is outwards from the center line, as I was telling you before. Okay, so how are we going to study how flies can detect the direction of motion of odor packets? And so if you go to slide 16, the approach that we took was to use optogenetics. And so this technology, what it amounts to is we can genetically modify the flies such that in their antenna, which they use to sense odor, in the neuron that detect the odor, they're called olfactory receptor neuron, we can express a protein that is sensitive to light, and that when it's activated opens a channel that brings current inside the neuron and activate those neurons. So you can activate the neuron with a virtual odor, which is light instead of real odors. And that's awesome because then light you can control with a projector. And so here on the right hand side, you see a picture of our rig where, so you have that big arena where the flies are walking, and on top of it, we're projecting a movie of those Odour Plumes that I was talking about before. And we're going to do this experiment in flies that are blind, so they cannot see that red light. Okay, and you can run control to show that basically the behavior of the flies in response to the light is very, very similar to that, to real odors and so on and so on. Okay, any question so far? Are you still with me?
I'm good, but let me check again. No, so far I
Okay.
think everyone is good, but it's a very elegant solution. Thank you.
So the great advantage of doing that is that we can dissociate the motion of the odor from the motion of the air. Okay, and so this is important. And so we can conduct experiment where there's no air motion, there's no flow, there's just odor motion without air motion. And so and that's the first actual result that is in the paper, and then I'm going to show you now. So if you go to slide number 18, so the first question we ask is, okay, so if the flies respond to the motion of the odor, we should be able to project red bars on this arena and have those bars moving from left to right or right to left. And it would be natural for the flies to turn against the motion, since they would be interesting to go towards the source of the odor, no? And so when the bars are moving from left to right, the fly should turn towards the left. And when the bars are moving from right to left, they should turn towards the right. And on the right-hand side of that slide, you can see that this is what's happening. Okay. Now, to get into a bit more details, what we did is we wonder, well, maybe they're also just sensing the gradient, no? So they have two antennae, and maybe when they hit the edge of one of those bars, one of the antenna is in the odor, the virtual odor, and the other one is not. So they know, oh, I should go to the left. So how do we know if they follow the gradient or the motion? And that's the next slide, the slide number 19. So the next experiment we did is that we took bars that were wider, and we slide those bars again on the arena. Now we project them as moving. So now when when the fly first experiences the bar, it's going from no odor to a large odor, no? And then when the other edge of that bar passes over the fly, it's the contrary. The odor is going from high to low. So if the fly, like you can see on the left of the slide, no? So if the fly is responding to the gradient, at the on edge in this picture, it should turn left, and at the off edge, it should turn right, because on the right is where the odor is. No? But if it's responding to the direction of motion, it should turn left on both sides, because the thing is moving from left to right. So what you can do is now basically project bars that are moving over the flies, and then you track also the flies, so you know the fly orientation relative to those bars. So then you can make a plot like the one on the right, where you have, if the fly is oriented perpendicular to the bar, basically there's no response because the two antennae receive the same thing at the same time. But if the fly is oriented heading parallel to the bar, then it should have maximum response. But the response should have a different sign for the on and off edge if the flies are sensing gradient, but it should be the same if they're sensing the direction of motion. Okay. And so if you go to the next slide, you can see the result of the experiment is very clearly showing that in both cases, the on edge and the off edge, the flies are responding in a very similar way. Any question about this?
I think so far it's good.
Okay.
So
let me continue.
So could both not be also possible? Like
that's a very good point. So of course we know that the flies also respond to gradient. Like if there's a smooth gradient, the flies will turn toward the side that has higher intensity. So it has to do probably with the temporal dynamics of the signal, and this is something we're examining right now. So the question we're asking now is like, how does the fly decide whether it should follow the gradient or the motion? And it probably depends on the statistics of the environment, no? And the amount of fluctuation and so on and so on. So here, this experiment was done with bars that were like very sharp edges, no? And so that seems to have promoted basically their response to the motion. So now the next question is, how can they tell this direction of motion? So what kind of computation? So that's on slide 21, I have that question. What is the computational algorithm of odor motion sensing? And so an obvious thing to do there is to say, well, is there any relationship between odor motion detection and visual motion detection? And motion detection in vision has been studied a lot. There's hundreds and hundreds of papers about that. No? And so now I invite you to go to the next slide where the title is What is Motion? And there's a movie 2. So if you load that movie 2 and play it, so when you play it, stare at the center of that flickering bars and tell me if they're moving towards the right or towards the left.
I don't know. For me, they're moving both ways, but it's
So
probably
they're flickering, but if you fixate on the center, so you should get the impression that they're moving to the right. But actually, it's an illusion. So if you go to the next slide, number 23, I show you how we generated this signal. So this signal basically consists of, on the plot on the right, you have in the horizontal direction, the position, and in the vertical direction, time, no? So if you take the first line there, you have a white, red, white, white, red, red, etc. No? So that would be what you see at a given time point. And so then for the next time point, what is projected is constructed in the following way. So if there was a red square, then there is a higher probability that in the next frame, on the right of it, there will be a red square. And if there was a white square, then there's a higher probability that on the right of it at the next frame, there's going to be a white square. And so you see that there's this kind of slanted directionality to the plot on the right, which plot the position of the bar as a function of time, no? It's kind of random, but there is some correlation. And so that signal doesn't have really net motion. What it has is just correlation. And our visual system detects that as motion because our visual system uses correlation to detect if things are moving to the right or to the left. And so if the flies use a similar type of algorithm as the visual system to detect motion, we should be able to project this signal that doesn't really have net motion, net direction, no? Of motion, just correlation on the flies that are walking around, and they should respond as if it was bars moving from the left to the right. So if you go to the slide 24, you can see that only pay attention first to the blue. I mean, I would have liked to animate all of those slides, but somehow it was impossible. So only pay attention to the blue curve. So in the blue case, we are basically having the bars have correlation towards the right. So the fictive motion is from left to right. And we also have wind in that experiment. And you see that the flies respond strongly with this the blue data there, showing that the flies are basically responding to the motion as before. No? But now we can do a new experiment that is impossible with real odor, where we put the apparent motion of the bars in the opposite direction of the wind. So the correlation now going from right to left rather than left to right. And so if the flies were still responding the same way, it would mean they don't care about those correlations. But you can see that clearly now the flies are very confused because the wind is going from left to right, but the correlations or the virtual motion, the illusion is going from right to left. And so you see that the response of the fly is kind of like reduced, okay? But we can go a step further. So there's another type of experiment you can do with humans and with flies in the visual system. And that's the next slide, the slide 25, where that has a black background. So there's two movies there, the movie 3 and the movie 4. In this case, we put the correlation still from left to right, but now we inverted the polarity. So whenever there's a dark square at a given time point, the next time step, this dark square, there's going to be a bright square on the right of it, okay? Or a higher probability to have. So the correlation is still from left to right, but the sign is changed, the phase is changed. And we, the human, detect that as reversing the direction. And you can test that on yourself. So if you look at the movie 3, if you open both movies next to each other, the movie 3 and the movie 4, you should see that the 3 is going from left to right and the movie 4 is going from right to left. But there's no real motion, that is just this correlation. Can you see that?
Yep. Yeah, I can see that. Thank you. Yeah, I think everyone also.
And so this is a very strong effect, and it's an illusion that's revealing how our visual system is actually computing motion. So if the fly olfactory system follows a similar principle to detect motion, in that case also just inverting the phase but keeping the direction of the correlation the same should also invert the response of the fly. If you go to slide 26, you see that it's exactly what happened. So the two experiments on the left are exactly the same, but now basically the phase of the correlation is reversed, okay? And you see that the two colors in the response and the experimental data are reversed. So basically, this really demonstrates that those flies are responding to these visual motion illusion, but with the olfactory system, okay? Any question?
No, it's really interesting that it works that way. Do Do you think it has to do with maybe I should ask later, but do you think it has to do with that when you showed with the vortices that the odor just expands, so it would be too unspecific of a signal?
Yes, I'm going to get there. So the next question, this is a good segue to my next question. So slide 27, the question then is now, okay, so we know now the flies can detect the motion of the odor, and they can do that even if they, independently from detecting wind. And they tend to respond by going against the motion. So the question then is, how is this odor motion detection useful for navigation? No? And to answer that question, we first have to go back to, so slide 28 is basically the same as the slide I was showing before, that shows that when you have odor released in a turbulent stream of air, imagine that the turbulence is totally homogeneous. So there's a mean wind and turbulence added to it. No? That can be totally generated, for example, in an experiment. So the mean wind direction is really from left to right, but the spreading of the odor is also from left to right, but also in addition to that, also away from the center line. No? And so if you detect the motion, you get information, a directional information that is additional to the one provided by the wind. And the hypothesis is that the odor motion indicates in which direction to go to find the center of the plume. And this is really important because remember, all the previous studies that I was talking about, including ours, concluded that basically animals, in order to navigate these odors, what they have to do is that whenever they detect odor, they go upwind. And when they lose the odor, they go crosswind to find the odor again. But here we're seeing that maybe they can do something better because there's not just the upwind direction they can detect. They can also detect the odor motion that indicates in which direction, in which crosswind direction to go to get towards the center of the plume, which the other strategy doesn't tell you anything about. So to test this hypothesis, we did the next experiment. That's slide 29. So there we basically put the fly in an environment where there's wind coming from the left, laminar wind, totally regular. And then we had bars, moving bars like before, but those bars were on the left, the bars were coming from the center line and moving outward. Where like on the right of this slide, this was a second experiment where the bars were actually moving towards the center line. So in a completely unnatural way, if you want. So on the left, the bar motion mimics, if you want, the natural spreading, dispersion of the odor away from the center line. But like on the right, it goes towards it, which is basically contrary to what happened normally in nature. And then on the bottom part of that slide, you see trajectories of flies navigating these two signals. And then the red box was what we considered our target. And you see that on the right, when the bars are moving in this unnatural way towards the center, the flies have a hard time getting to the red box. And you can see that while they are in the back of the arena, they're kind of moving in the same way in both experiments. But when they reach that triangular tapering tip of the plume, no, they get lost on the right. Where like on the left, they're able to follow it towards to the box. And if you look on the bottom right of the slide, I plotted the distribution of the position of the fly in the right half of the arena and the left half of the arena, and see that in the left half, for the purple, which is where in the case in which the bars are moving in a natural way, there's a much more the flies are concentrated much more closer to the center line. And the success rate is plotted above. You see there's a significant difference. So that really suggested that actually this odor motion is useful even in navigation. But so then to really convince ourselves, we did the next experiment, which is actually, I thought, is beautiful in some way, because what we did there, so this is slide number 30, we took one of those smoke plume that we had imaged with our camera, and we projected that pattern. So you can look, if you download the movie number 5 and you play it, you can see that Odour plume. So it's good on a gray scale. No? You see the things coming from the left and basically moving towards the right. No? But on the right side, we play the same plume, but reverse in time. So we play the movie backwards. It's exactly the same movie. We do an experiment where we play the movie in the normal way, and we do another experiment where you play the movie backwards in time. If you play backwards in time, what you're reversing is all the correlation. So the motion is reversed. But the gradients, the concentration, and so on at a given position, on average, they're exactly the same. It's the same movie. It's the same frames. And so that should enable you to test whether the odor motion really affects navigation. And so if you go to, I don't know if you had a chance to check out the movies, but you should be able to. So if we go to slide number 31, this is the result. You see that on the left, where we're playing the movie in the normal way, the insects are moving to the box in greater quantity than when we play the movie in reverse time. So in these two plots, the gray lines are the fly's trajectory that didn't make it to the target, and the white lines are the trajectory that did arrive at the target. And then below you have again the success rate. You see that is much higher for the flies responding to the movie played in a normal way than when we play the movie in reverse. And the only thing really that changed is that we're playing back in time. So. And so that really shows that the flies are actually using this information from odor motion, because that's what's really reverse when we do, where we play the thing backwards in order to navigate. And that's basically how we finish the paper. We also run simulation with simulated robots and stuff like that. But basically, this is like the main result. So that brings me to my take-home message, slide 32. So what we discover is that the olfactory system can detect the motion of odor packets. And that's a sense that, nobody knew about, even though we've been studying flies' behavior for a very long time. Odor motion detection seems to be obeying a correlation-based type of algorithm, which seems to be very similar or equivalent to visual motion detection algorithm that we know exist in humans, in the flies, etc. And we also demonstrated that basically odor motion seems to provide a directional cue that is complementary to that of the wind, because it points towards the center line of the plume. And so that helps the animal that's navigating stay near the center of the plume and not lose the plume. It should enhance basically performance in navigation. And flies really exploit this odor motion during olfactory navigation. So now I'd like to thank the people who did the work. Really, this was totally spearheaded by Nirag. And Mahmoud is the person who designed the experiment with the smoke and so on. And then Nirag, when he came to the lab, he basically added the projector and then used the same setup. And the reason Nirag added the projector is because our neighbor is Damon Clark, and Damon had developed this projector system to study the walking behavior of flies. And he has been a long-time collaborator of us, studying also olfaction with us in previous paper, including the one with Mahmoud Demir. And so this was really possible, I think, because the two labs are next to each other in a very interdisciplinary environment, in a place called the Quantitative Biology Institute. And the people from Damon's lab who study motion are sitting next to the people in my lab who study olfaction. And so together they put 2 plus 2 equal 4, and that's how they got to discover this. So thank you for your attention, and I'm happy to take questions. And
Thank you so much for this wonderful presentation. That is so interesting, and also that you shared this collaboration environment that made this possible. That's really inspiring, I think. And yeah, I wish, everywhere it was like that. So, mm, so it's really interesting how this motion is really important. And you said that was probably very helpful that you studied physics before that. So when you simulate this with robots, or do you assume that mammals like have a similar way of perceiving odor, or is it especially important for flying, animals?
mm-hmm. Yeah, so this is this. So one of the speculation we make at the end of the paper is that basically we expect that many animals will actually detect odor motion, because the only thing really you need to have to do that is that you need to have two spatially separated sensors, and then do a comparison in the correlation of the signal between the two sides. And so there's really no reason why humans and dogs and moths and you know even fish and so on uh, will not exploit that that possibility to detect the motion of those signals. And in terms of the robot, what we did is that we simulated robots that so there's another part in the paper where we show also how do the animals combine air motion. It's known that the flies turn upwind, no? They respond to the direction of the wind also. So we wonder, how do they respond to the direction of the wind and the motion of the odor? And so the first thing that we looked at is if they were basically just summing up together those two directions. And we found that actually that's what they do. So to a zero order, what seems to be happening is that the flies turn against the direction that would be the sum of the direction of the wind and the direction of the odor. Okay? And so when we simulated robots, we first took robots that basically performed the older algorithm, which is they would turn upwind when they detect odor and crosswind when they don't. And then we added to the algorithm the fact that they would turn not against upwind, but against the sum of the direction of the wind and that of the odor motion. And in all cases, we found that the robots are doing better.
So that's interesting. Is it known maybe in people or animals that have kind of a deficiency in odor sensing that their navigation in general is a little bit off, or
I don't
has
know.
that?
That's an interesting question. I don't know, but maybe somebody in the audience knows. I don't know. But yes, so I know that there's some paper about human detecting um, if an odor arrives first on the right or on the left and things like that. So there's some papers that have been published about that. There's also some paper where people have studied how shock respond to whether the signal appears first on the right nostril or the left nostril. And again, they find a response to the timing of the odor, you know, if it comes first on the right or on the left. And so I expect that, you know, there's going to be now a lot of studies that look specifically at, you know, signal like the ones we've been using. You know? If you can make a signal that has just correlations, do the animal respond to those correlations?
I mean, we were all stuck at home during COVID, but that would have been a great time to test it.
Yes, yes.
I mean, um, yeah, the other thing is, what's while we're eating or like an animal is eating, it's also sensing smell and stuff. If you would
Mhm.
additionally add another odor that, you know, comes with the motion, would that be kind of off, or can we add layers of information in odor, or can we just focus on one basically?
Yeah, so the olfactory system is able to detect new odor on top of odors that are already in the background. And so we have that ability that, you know, you smelling something in the environment, like in a room. Imagine that you're in a room where somebody smoked before and you smell it, no? But then there's somebody that passed by with some perfume and you're going to detect the perfume. So you can detect on top of other odors and differentiate one from the other. There's also a lot of experiment. There's some experiment showing that animals can detect if molecules came from the same source of odor or not. So if there's a source of odor that releases, like, let's say, 5 compounds, odorant compounds, then these are transported by the air in a correlated manner, no? In a synchronous kind of correlated manner. And so the temporal structure of the arrival of those different components of the odor really matters for the animal to decide, oh, that's an odor object, if you want. There's a source there that contains that odor. You know?
Yeah, thank you. I'll ask one more question and then I'll hand it over to Dr. Shah. So let's say we want to build a robot that's humanoid type of, or dog type, or whatever, Boston Dynamics.
Mm-hmm.
And we are not good at making artificial odor sensing. So, but let's say we would be good at it and we would have this different inputs and stuff. But are the current neural networks, like the artificial ones, would they be able to do that, to sense all these different odors, you know, with the
Yeah,
navigation?
yeah. So people are developing robot that, or there's already a robot out there that can detect odorant and navigate odorant. Um, One of the big difficulty was having a good detector for odors. So one way to detect odor is to suck air into what is called a photoionization detector. So you pass the air between two plates and you ionize the gas, and then that gives you a voltage difference, and then that you can build the system such that the voltage difference is linear with the concentration of molecules of odor. But these instrument tends to be kind of bulky and they suck air and so on and so on. But so there's been like development of miniaturized electronic component now that can actually detect odors. And so there are robots that are being built where, you know, you have, but so far I think all the robots that have been built, they basically detect the time dependency of the odor, maybe the gradient of the odor. So they have two detectors and they can tell if it's more intense on the right than to the left. But no one really has used yet the fact that one could also compute the motion. And so this is something that's, right to be done right now. Like if you have a company building robots that smell odor, what you should definitely do is add odor motion sensing to the algorithm that the that the robot is using. And then um, the strategy that's being used by the robot to navigate, given that the robot is getting the information about the concentration, the gradient, and maybe now in the future the motion, um, that is still like very much open, a subject of open research, current research. Like actually, our lab is studying what are the type of optimal strategy that an animal should use to use that information to reach the goal the fastest, you know? And so that's also very interesting. And there, machine learning and so on is being used to study that question.
Yeah, that's interesting. Thank you. Because I think adding all these senses is really interesting for someday having an AGI. I don't think you can really achieve that without adding more. I know like the OpenAI chat function is getting really good, but that's just one type of
Yeah.
information input, so I don't think that's enough. But Dr. Shah, I'm sorry that I asked so many questions. Go ahead. Thank you.
No, thank you so much. So that was very wonderful research, and my question is in continuation of the Katerina question about the AI, because the practical level of use of your research, for example, it can be for the detection of the poisonous gas. I don't know that
Yeah.
if you ever thought about it. Also, you just talked about the photon and impact of the light, and I was just wondering, did you ever think about the artificial light and having this impact in during the night time?
You mean I didn't understand the question about the light. So
Yeah, you talk
you
about the
you mean
photon, and I was just wondering if we want to consider, for example, I mean, this kind of phenomenon that is happening during for example, day or night, and by considering the artificial light, do you think that, for example, with producing the chips or those kind of things by, uh, you know,
so
those
so
kind of,
in
because you talk about
yeah,
robot and all of those things, and
yeah,
my
yeah.
idea is about the chip, and I was just wondering maybe you going through deeply
Hmm.
about this.
So like so first in the experiment we use light, but that's because we're using optogenetics now. But we have also experiment with real odor where we so that you cannot see, but we know where the odor is, and we can show that the animal is responding to the motion of those odors. And so, uh, in terms of navigating odors, it will make no difference if it's during the day or the night, in the sense that motion detection will basically just involve detecting uh, the odor in that case. And I don't know if that answered your question.
Okay, and about the poisonous, I mean, gas
Oh, so
detection.
yeah, so that's definitely something that so those robots that are being built that can follow odorant, they're built to detect bombs and artifacts like that. They're built to navigate in an environment where humans cannot go because they're too dangerous, etc. No? And those robots that exist are already have some olfactory capabilities, but they don't have the capability to detect the odor motion. So that is clearly a place where, one could, you know, improve the performance of those robots quite a lot, I think.
So
Yeah.
think about the fly. and odor is one side, and another side, for example, it can be different in sex, and we can think about some hormones like a pheromone. And when we want to just compare them to each other, maybe it's not comparable in some ways, but still it can provide some spatial data, and they can use it for the AI. That's why I ask you about the differences between the day and light and how it can work for the colonies. For example, when you're talking about the fly, they are not living in a colony, but some other insects, they are living in the colonies. And I was just wondering, because this is
I see.
your
So
research.
so you mean if like for example animals are moving as a group,
Yeah,
as a
it can
colony?
be,
That's
for
what
example,
that's yeah,
bees,
yeah,
right? Uh,
yeah, yeah,
just
yeah, yeah.
insects like bees,
This
not.
yes, that's very interesting. So um I I haven't thought about what would happen to other motion if, you know, you have a close by other animal that is also flapping its wings, for example. And but in general, what I think is so in the case of the bees, for example, no, the individual bee goes and explore, no, and try to find like um, some sugar somewhere, and then find some flowers that are, you know, the one that it likes. So then it goes back to the hive to basically tell the other bees to go in that direction. And in the navigation of the bees towards the source, so then a whole bunch of bees will try to fly towards that source, no? And then at some point during that navigation, they are going to use the olfactory system and to detect the odor that are being released by the source, by the flower, and so on. And so in this case, they're going to be able to use odor motion, definitely. Another thing that is interesting is that um, maybe you can also use odor motion if you know your own speed and there is the odor landscape is kind of static, but you moving in that landscape, then you get an odor motion that is due to your own motion within the landscape. No? And so that's another thing we kind of currently studying. So the odor motion really is relative to your own frame of reference. And so it could, if the animal is moving very fast or moving slow, its perception of the motion is different. The same with its perception of the wind. And so in that sense, being in a colony maybe also will change things. But I haven't thought deeper about that so
Yeah,
far.
it can be a speed, and in the same time, you mentioned about the signaling process and the protein, because I'm assuming you don't
Hmm.
have any information right now about the genomic data or such a things. But I mean that about this specific research by itself, because you didn't mention during the slides. But I was just wondering about the effect of the temperature, because they have a thermoregulatory process, especially
Hmm.
is come along with detection of the pheromone, or it can be other even. So that was just out of curiosity to ask you, because you know better about that.
Yeah, yeah, yeah. So temperature definitely should affect the olfactory, basically the sensitivity to odor. And if the sensitivity to the odor signal is reduced, then the ability to detect the motion of the odor is also changed, you know. So we do expect that there would be like a change due to the temperature. And what was the other part of the question?
Yeah,
I'm blanking now.
exactly. It was a
Oh, about
thermoregulatory
the
inside the insect.
yeah, it was also about the mechanism, no? So we currently trying to find what is the circuit in the brain that actually compute this odor motion. So that's one thing. And the other thing that yeah, I mean that's what we're trying to do. So we want to first identify what is the neural circuitry inside the brain that is used to detect odor motion, and then compare that to the one that is known to, in the fly brain to use to detect visual motion, and see if there are similarities, differences, and so on. Because it's kind of interesting that maybe these two motion detector have evolved, twice. Like one for olfaction and one for vision. So we're going to be able to compare the two.
Yeah, that's interesting. Also, the speed, right? If there's a difference,
Mm-hmm.
because the visual system is pretty fast. Transmission, although it's a pretty long path, like especially in mammals and so on.
Mm-hmm.
The audio one is even faster, but it just has less connections. That's why it's faster. But the speed of the the signal transduction itself, I don't think is
So
fast.
in
It's
in
less. Yeah.
so in the fly, the fly olfactory system is also very shallow. So there's just a few synapses between the periphery, the sensory, basically the receptors, and the brain, no, the cortex. And so um, and one thing that is in the paper, so what we can do is calculate. So how does the you know how do you detect motion from having two sensors that are separated spatially? No, so there is a general a general model for that is that imagine there's a left sensor and a right sensor, no, and that an object is coming from left to right. If the signal downstream of the left sensor is delayed, but the signal downstream of the right sensor is not delayed, then if the object is coming from left to right, it will hit the left sensor, but that signal is delayed. Then it hits the right sensor, but that one is not delayed. And so if those two signals then coincide and are kind of multiplied or nonlinear interact, you're going to get some positive signal that would say, oh, something is moving from left to right. But the same exact two neurons, if an object had been moving from right to left, it would hit the right sensor first, but downstream of the right sensor, there's no delay. And so the signal will go down, and then later on, it hit the left sensor, and now there's a delay, so they will not coincide downstream. And so you will not get a signal. And so this architecture is called the Eisenstadt-Raycart correlator, and is basically the basic principle of how motion is detected in the visual system. And what is critical there is that delay. And so we did an experiment where we actually measured that delay for the olfactory system, assuming that the olfactory system would be working under a similar principle of having two input, but one is delayed and the other one is not. And we found that the delay that comes out of the data is on the order of 20 milliseconds, which is exactly in the same ballpark as the one that's being measured in the visual system. So that's interesting. The
Yeah.
similar time scale.
Oh, that's interesting. And then is there also a lot of feedback inhibition because the visual system has a lot because I think it's to keep the information quite granular. Um, would be interesting also to compare like how
Mm-hmm.
but like how detailed the information will get passed on because I think by just looking at how many different GABA receptors and autoreceptors are there. Maybe just screening that would give you already a kind of a hint.
So
If
we kind of investigating um, So we can, with the fly, the cool thing about it is that we can study the response of the fly if all of the olfactory receptor neurons are activated or just one type is activated. And so we're doing that, exactly that currently to kind of try to answer the question you're asking. Yeah. So that's
So it's kind of
I don't know the
different
answer.
than than the ganglia, because we are just considering one ganglia for insects, and we should not think about afferent or efferent technique based upon whatever you are saying. This is just one direction, and for example, we have to exit arrows for the olfactory based upon whatever you are explaining. But as a fact, that's a one ganglia, right? In insects normally working.
So in the olfactory system, basically you have every olfactory receptor neuron only express one type of receptor, but then all the neurons that express the same receptor, they project to the same region of the brain called glomeruli, and there they make synaptic connection with the second order neuron called projection neuron that then project to downstream to the brain. No? And already at that level of the antennal lobe where you have those glomeruli, you have local neuron that basically will inhibit. So if one of those projection neuron is activated, the other one tend to be inhibited. And so uh, at that level already, you basically have signal that come from multiple channels at the same time. And so the difference with the vision is that in olfaction, there is very large number of receptors. No, there's like families of genes. That actually is some of the largest gene family in the fly genome that code for different type of receptors. And so you can think about the olfactory system as like having, let's say, 50 different channels. And one odor molecule arrives and it will interact with channel 1, 3, and 7, for example. No? And so you can think about this 1, 3, 7 as the combinatorial code that identify that odor. And then another odor arrives and it might interact with channel 1, 23, and 50. And so you have this additional problem that the olfactory system has is to basically detect the identity of the odor molecule in addition to detect temporal change in the concentration of all of those molecules. And you have an enormous space of possible molecule that those systems have to be able to detect and make decision about in a very, very quick manner.
as
And
as well as the memory, if it want to keep the
and
memory of
memory,
that or
yeah,
not.
yeah.
Okay,
And so
thank you.
a big a very interesting question in olfaction is how can the signal, two things about the signal have to be encoded simultaneously and very quickly. The identity of the signal is not just red, blue, and so on, but it could be like, out of 1,000 or out of 10,000, and the concentration, which will give you correlation, the detection of gradient, motion, etc. And so in our lab in the past, we've published a bunch of paper showing that one thing that's really interesting is that the olfactory receptor neuron can encode the timing of arrival of the odor super precisely and independently from the concentration of the odor. So if you calculate the cross correlation between this odor signal and the olfactory receptor neuron responses, you find that that cross correlation is actually independent, at least over a certain range, over of the concentration of the odor, which is kind of a amazing thing. But it makes sense because if you're flying around, you know you want to detect those packets of odor. You want to know when they arrive, irrespective of the concentration. You know? And to detect the motion, you want to extract information about the motion, irrespective of the concentration of those packets. And so timing has to be detected very precisely.
Yeah, and then you want to, you know, look back and find it again, probably. So that's also at the same time. Like, you want to find the source of good food. So imitating them would make really good cooking robots. I'm kidding. Probably
Yeah.
better than human cooks. I don't know.
So, I mean, I don't want to, take too much of your time, but it's like 6:30 almost, so
Yeah, I know.
I've talked a lot.
Yes, I wanted to check in with you. We have Jake and Dennis. Do you have two more questions available? It's
Sure.
fine if you say no. It's
Yeah,
totally
yeah, absolutely.
okay. Jake and Dennis, thank you for waiting.
Hi, thanks. Yeah, I was thinking about mosquitoes, and the most effective mosquito traps use carbon dioxide, like a carbon dioxide source,
Hmm.
to trap mosquitoes. And so I'm wondering if you ever thought about using your techniques to figure out a way of using visual cues to attract a mosquito and making better, more accessible mosquito traps.
Yeah, I think that so one thing with respect to that is that um, when animals like mosquito try to reach their target, they use different modalities depending on the distance. So when they're really far away from the target, they really just use olfaction to to track the target, no, to get to the target. When they get really close, then they engage the visual system. And there's an experiment by Michael Dickinson, for example, at Caltech, has shown that not with mosquito but with flies that this is the case. So as they approach closer to the source, then they get very sensitive to contrast and shape of objects and bright lights and so on. And so definitely, I think that that's something that might already exist or could be engineered, I think.
Very cool, thanks.
Denise, did you have a question? Hey, how are you?
Hi, everybody. Yeah, just curious. Was there anything that you wanted to necessarily room in the paper? It's sort of adjacent, but not necessarily in scope for this particular thing. Any that didn't really quite make it in here, but you think would be interesting for folks?
Oh, uh, I mean, yes. I mean, there are like some, um, let me think. I, I, the thing that didn't make it in the paper, and so, but also, when we were writing the paper, we really focused on the thing that would go in, and so we kind of have preliminary result, and we're going to pursue them, but um, we would to put them in the paper, we'd have to do more work. So that's why I also didn't make it in the paper. But that are related to, for example, the strategies to use to navigate to the source, which we're investigating now using machine learning and so on and so on. And another aspect was what is the neural circuitry that um, um, the olfactory system is using to compute motion. And then also we have more data about under what condition, what are the failure mode of odor motion. So, there's a range of odor speed and wind speed over which that works, and beyond which then um, it becomes impossible to detect because the delay between the two antennae is too short or too long. And in general, um, we were also very interested in understanding how the statistics of the Odour plume, which are different in different, if you have a source at a certain position, as a function of distance from that source, the statistics of the turbulent Odour plume will change, no? And so we're interested in how basically the animal will adapt its navigation as it's going to the source. But I mean, this is really just going beyond that paper at this point.
Yeah, thank
Fascinating.
you.
As someone who has studied aerosol science, that was a really interesting. Um, so
Oh,
thank you
thank
so
you.
much. Anyone else have questions?
Yeah, I know that Kyle and Philip joined. Just be really quick because we wanted to stop like in a minute. So thank you.
Yeah, I'll be incredibly quick. Thank you. From what I was able to listen to, it was amazing. I'm curious, have you worked with anybody on mindfulness? Because listening to you, it sounds like you could be a very valuable person for them to help with sensing odor, and I know, and navigation as well. And I understand this is a little bit outside of the scope of your work, but I intuitively feel like if you were to approach somebody that's working on mindfulness, that your work on odors and navigation might actually contribute.
Oh, that's interesting. So, a, a few years ago, when I was not yet studying the motion of the odor, but basically just olfaction, um, I got interested in the fact that it seems that odor therapy is kind of used um, for example, treat some aspects of autism, and basically odors, so the olfactory system is basically one of the really most ancient sensory system that animals have, no? And it's also very shallow in terms of like there's very short distance between the sensor and the brain. And so a lot of you probably have had this experience of like you smell something and then you remember things in your childhood and so on. And so in that sense, um, I've thought about, uh, it's related to mindfulness in some sense, but I definitely think that olfaction is a sense that affects our moods, our sense of perception of well-being and so on in ways that are not fully understood. And it's not it's on the dark side, it's not like a random, the fact that advertisers have realized that you can attract people into a shop by releasing odors that are attractive. And so, you have this kind of fake odors of bread and stuff like that that people use to bring people in in a bakery, etc. No? So it's a very interesting uh, question, but I don't know much about it, but it's interesting. Thank you for the question.
Yeah, Yeah, that was amazing because you even brought up the scent and episodic memory there.
Hmm.
Like it seems like people come and encounter a scent and then all of a sudden be presented with a memory, an episodic memory from their childhood or something. So amazing
Yeah,
presentation. Thank you so much.
thank you.
Philip, you have the last quick question. Thank
Thank
you.
you, thank you, host, and thank you, Terry. Terry, I hope I didn't butcher your name. Um, I am a beekeeper, Apris, and basically what you were speaking of was very relevant to what I am observing through my several years of practical beekeeping. Um, I am trying to develop. I see from my observation. I'm a Sankofa scientist, meaning it's an African ethos of retrieve, repair, and restore. So it's a self-exploration tool, or a self-navigation tool, because you're talking about the said
Hmm.
thing. What I wanted to say, I don't want to speak too far, but I do want to develop. I am interested in, from my observation, I have seen a correlation that needs to be um, developed in relation with the odor therapy. So I'm proposing, and maybe some of the others in this group have heard of it already, apotherapy, which in particular is the purified or the microclimate air within a healthy beehive, and to use that
Mm.
as an aid to increase the what we say, the good brain gut bacteria, so that um, respective recipients can develop a greater ability or recovery or regenerative, quality of life, whether it be
Hmm.
short or long. So thank you. So I don't want to speak too long, but I did request you guys to follow, and hopefully I can have another conversation with you guys and others who are interested. I noticed Jake showing a beehive. Respect, more love.
Thank you. That's really interesting. Especially because in my lab we also work on bacteria, and a lot of the odors that people have and so on are actually generated by the bacteria. So the smell of your skin is really the smell of the bacteria on your skin,
Very true.
and
Very true.
and so also I imagine in a beehive, uh, a lot of the smell of the beehive has to do with also the microbiome
Yes.
of the flies and so on. So yeah, it makes sense.
Yes, and the plants that they interact with,
Yeah.
the microbiota of those plants and etc., which obviously links back to the memory. Obviously, not the long-term memory like they store, but a memory that they are able to retrieve, more accessible, based upon the microbiotic environment that they are
Hmm.
connected to. Thank you very much. As I said, I hope to follow this on, have a conversation about that development of the odor therapy and seeing how it can aid people with learning difficulties, depression, obviously mental health, obviously since the lockdown and COVID, long COVID. I believe that it could aid in long COVID. There's other evidences of it can help reverse. Oh, there's loads of things. But anyway, thank you very much. I won't stop speaking for now. Thank you.
Thank you.
Yeah, thank you so much, Philip. That was really interesting. And yeah, so thank you so much, Thierry. This was such a wonderful talk and then discussion also. So we really appreciate you coming here and taking the time to talk with us. And I learned a lot. So, and I think everyone appreciates it very much. And
Hmm.
yeah, maybe one day you come back and give us updates, maybe next year around this time. That would be wonderful to continue learning what you learn in the lab. So thank
Sure.
you.
Um,
Thank you very much for having me. Really, it was like, um, you know, I don't get a lot of chance to talk to such a wide open audience. And the only frustration, I would love to see you in person instead of just talking like this. But also, I realized that in this way, you can talk to people all over the places, no? So, and if you have any question, don't hesitate. You can email me, and I'll try to answer. Okay.
Yeah, thank you for saying that. Yeah, if you need the contact, if you can, it's the I put the web site of your lab in the chat, so
Mm-hmm.
for sure there for everyone. And I agree, you would love to meet also in person, but yeah, that way we can reach people around the
Yeah.
world, and maybe one day we'll have a place to meet where people can come.
Okay.
Maybe if I become a millionaire one day. No, I'm kidding. Okay. Okay. Thank you, everyone. Enjoy the rest of your day. If you like discussions like this, you probably already heard this. Just follow the club, and we'll have tomorrow a talk about the Hipparcos star catalog, how it was revealed, which will be really interesting. And then on Friday, we'll have actually a microbiota talk, and how it promotes social behavior and through microglia interaction, which is really interesting. I haven't seen that before, and I'm really looking forward to it. So I hope I hear you all back again. And Thierry, thank you so much for um, gifting us your time and knowledge.
Thank you for the invitation. Thanks
Okay.
to everyone for listening.
Yeah, and thanks for the great questions, everyone. Thank you. Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Bye bye.